Good morning guys. Today's topic is blood clotting. It is also called hemostasis. Once our blood vessel cut during some accidents or injuries, blood stream turns its flow out of its direction. Then it's called bleeding. Such a bleeding takes place minimum 3 to maximum 8 minutes according to the normal injuries. After 8 minutes, the blood flowing, nothing but bleeding occurs, then we have to suspect we have some related blood clotting factors absence, also some genetic disorders like hemophilia and anemia. How this phenomenon takes place after an injury? Let us see. But don't think that this process is a very simple process. So many factors and steps are involved in this process. Certain factors are required to perform all these serial reactions or cascade mechanisms during the blood clotting phenomenon along with the damage. If we observe all the 13 factors, they are damn required for the blood clotting process one by one. The very first one is fibrinogen. Fibrinogen is present inside the plasma, but that is synthesized in liver. Means it is produced by the liver and it is existing in plasma. Fibrinogen is inactive form. So even though all these factors, important factors for the blood clotting present inside the blood never clot unless blood vessel damage. Why? All these are existing inside the blood in inactive stage. Once trauma happened, trauma means damage of blood vessel, then all these can be activated. Let us see step by step later. Second thing is prothrombin. Prothrombin is a very important blood clotting factor. It is also synthesized inside the liver and existing in blood plasma. Third one is thromboplastin. Thromboplastin is another protein which is produced by damaged blood vessel wall and as well as blood platelets. I repeat, the third important factor in the blood clotting process is thromboplastin. Source of thromboplastin is two. One is damaged blood vessel wall also produces, and thrombocytes, nothing but blood platelets, are also releases this protein. Even the source is different, chemical is same. Fourth one, calcium ions. There is no doubt to say calcium ions plays a very vital role in the process of blood clotting. The source of calcium is bone. So excess amount of calcium from the bone released into the blood. So this calcium helps to form the prothrombin. Next one is proaxelrin and also proconvertin. This axelrin and convertin, all these are accessory factors. Means proaxelrin is labile factor and proconvertin is stable factor. So to accelerate the reactions, to maintain the stability of components required for the blood clotting, these two are cofactors. Next, anti-hemophilic factor, plasma thromboplastin component. It is also called Christmas factor. Ninth factor is also called Christmas factor. Last 10th one is a Stuart factor. Next 11th one is plasma thromboplastin antecedent. Next, Hageman's factor. Hageman is a scientist who discovered that, and fibrin stabilizing factor. Just you have to observe, I said 13 factors, here one is missed. You see 1, 2, 3, 4, 5. I written over here 7, means continued. 7, 8, 9, 10, 11, 12, 13, where is 6? Before means some decades back, the factors list, the sixth factor was also there. As these functions are unknown, it has been removed. So as their numbers are pre-owned, they have been continued without factor 6. So factor 6 is unknown. Factor 1 fibrinogen, factor 2 prothrombin, factor 3 thromboplastin, factor 4 calcium ions, factor 5 proaxelrin, factor 7 there is no sixth factor. Factor 7 is proconvertin, 8th factor is anti-hemophilic factor, 9th factor is Christmas factor, is also called plasma thromboplastin component, 10th factor is Stuart factor, 11th factor is plasma thromboplastin antecedent, 12th factor is Hageman's factor, and 13th factor is fibrin stabilizing factor. almost of all these factors except 13, all these factors helps to stabilize the inactive prothrombin into thrombin. Means before first step only all they have been used. Let us see the steps involved in the hemostasis process. Steps involved in the hemostasis process of blood clotting is also called hemostasis, prevention of blood clotting. Hemo means blood, stasis means control. Now there were 3 steps are involved, but everyone should know that generally a blood vessel inside blood flows in its own direction, right? Once this blood vessel cuts or damaged, you just take one side, damage, then the blood flow or components present in the blood comes out through this damaged blood vessel wall. So this process is called bleeding. When bleeding occurs, when blood vessel wall damages, bleeding occurs. Suppose this is the blood vessel wall, it cuts, then it is called a trauma. Then blood flow generally present in its stream in the one direction, it comes out. That is called bleeding. Once the bleeding occurs, generally what happens, the very first one is vascular spasm. Means the edges where blood vessel wall broken can be constricted generally. This is two, if you assume these two are edges of the blood vessel where trauma taken place, means a cut happened, then these two generally constricted. Clear? So that is called vascular spasm. So vascular spasm constrict the area so that flow of blood decreases but not stops. Second one, blood platelet plug formation. So all blood platelets in the bloodstream comes over here and form a plug so that blood flow means the direction, blood bleeding again decreases. By these two processes, blood flow which is going out of the bloodstream or out of the blood vessel decreases but not stops. Now very final step, that is blood clotting. Blood clotting takes place by series of reactions like A, B, C steps. The reaction A is carried out by so many factors. Then if reaction A happened, then reaction B will start. Once the B reaction happened, then C reaction can be initiated. So C must happen after B. B must happen after A. This type of reaction phenomenon is called cascade mechanism. Cascade mechanism. Without one reaction, its further reaction never takes place. So series of reactions are there. Each reaction is depends upon its before reactions. So that is called cascade mechanisms. Let me tell you again. Once blood bleeding takes place, very firstly vascular spasms. Vascular spasms do not require any reactions. It is a very natural process. The muscular contraction of the blood vessel helps to constrict the area. So when constrict area shorts, then flow decreases but not stops, you have to understand. Secondly, blood platelet plug formation. After vascular spasms, so many blood platelets comes closer and form a cluster like structure, that is called blood platelet plug formation. It helps as a plug. Thirdly, blood clotting. So blood clotting have so many steps. Let us see in detail. Once a trauma happened, if you see, this is the damaged blood vessel wall, right? Damaged blood vessel wall. From the damaged blood vessel wall, a chemical will be released. I already told you that is thromboplastin. Thromboplastin will be released by the damaged blood vessel wall. At the same time, inside so many blood platelets are also there. They are also called thrombocytes. these thrombocytes are also releasing the same chemical, that is thromboplastin. Thromboplastin is a protein which can be released by thrombocytes near the damaged blood vessel part. At the same time, blood vessel tissue, broken part, is also can be released thromboplastin. I already told you, chemically these two are same, but source is different. Means it is from internal source, means cells are releasing. That is why it is called intrinsic path. Intrinsic path. And this is extrinsic path. Extrinsic path means what? Production of thromboplastin from damaged blood vessel wall. Intrinsic path means what? Production of thromboplastin from thrombocytes. So these two are accumulating in the blood and converted into an enzyme called thrombokinase. Thrombokinase. For the production of this thrombokinase, calcium ions are very much required. At the same time, so many factors like 7, 8, 9, 10, 11, 12, all are required in the intrinsic path and extrinsic path. Finally, result is thrombokinase. Thrombokinase is an enzyme. This is the step 1, I can say. In the step 1, what happened? Thromboplastin has been produced by both the sources, extrinsic sources and intrinsic sources. Accumulated means from the both the sources, thromboplastin protein is releasing into the blood. So in the blood, thromboplastin levels will be very much increased after the cut or injury. So that increased thromboplastin under so many factors by the help of calcium ions converted into enzyme. So proteins converted into an enzyme. That enzyme is called thrombokinase. This is the first step. This thrombokinase, again in the presence of calcium ions, converts one inactive form into active form. What is that inactive form here? That is prothrombin. Let us see here. Prothrombin. Prothrombin is inactive form. So this prothrombin, which is inactive form, converted into active form called thrombin. Thrombin is active form. Who required to convert the prothrombin into thrombin? Thrombokinase is required in the presence of calcium ions. Clear? So thrombokinase, without thrombokinase, prothrombin cannot convert into thrombin. I can say this is step 2. In step 2, what happened? Inactive prothrombin protein converts into activated form called thrombin. So this is activated form. So second reaction happened because of thrombokinase. Thrombokinase happened because of step 1. So without step 1 there is no step 2. That's why I mentioned here all these reactions are called cascade reactions. Next, once this thrombin formed, in the presence of thrombin, inactive fibrinogen become fibrin. Fibrinogen, which is inactive form. This fibrinogen, under the action of thrombin, again here calcium ions are required. So that I told you calcium ions actions at many places in the phenomenon. This fibrinogen become active form called fibrin. Okay, this is step 3. In step 3, what happened? Inactivated fibrinogen become activated protein called fibrin. Fibrin is a mesh-like structure. It consists of so many fibrin filaments, and fibrin stabilizing factor, which is a 13 factor you just observe, helps to form this fibrin mesh here. So that complete stoppage of bleeding. Once a fibrin formed, this fibrin acts as a mesh-like structure. Along with the mesh-like structure, RBC, WBC and some damaged and some protein-like structures and also other elements of blood comes and cope up with this mesh-like structure and form a bridge like a barrier so that a blood flow cannot comes out of the blood vessel and eventually it becomes stoppage of bleeding. Stoppage of bleeding is called hemostasis or stoppage of bleeding is called blood clotting phenomenon. So this is how 3 steps are important. 13 factors are also important for the blood clotting in the span of 3 to 8 minutes. Let me explain again. Step 1, step 2 and step 3. Result, fibrin. Once fibrin formed, bleeding can be stopped permanently. If any one of the 13 factors are lagging in our body, does it happen? Yes. All these factors are proteins. Proteins are coming from gene. Gene is nothing but DNA sequences. In our body, any genetical errors are present and one or many of these proteins cannot be produced. Listen carefully. All these 3 factors are important as you have observed. Right? Fibrinogen is not there, then how fibrin will come? Prothrombin is not there, then how thrombin will come? Thromboplastin is not there, then how enzyme thrombokinase will come? Like the calcium ions are not there, how all these reactions takes place? Other than this, all rest of these elements are important for intermediate. So, if any factor is absent, how it can be absent in our body is your question. I can say, each and everything is a protein. Protein is coming from mRNA. mRNA is coming from DNA. DNA is nothing but gene. So, sequences, they helps to produce a particular protein were absent, genetical disorders, then one of this list or many of this list proteins can be absent in our body. If they are absent, these reactions cannot be carried out. If they are not carried out, the bleeding time will increase, called hemophilia. So hemophilia is a kind of autosomal disorder. Clear? In second year genetics lesson, we will continue this part, not now. So this lesson helps to get some basics of the circulatory system to understand blood clotting mechanism. Let me review entire the concept. Blood clotting mechanism is also called hemostasis, in which 3 steps are involved. Firstly, vascular spasms, in which blood flow decreases due to the contraction of blood vessel wall which is damaged. Number 2 is formation of plug by the blood platelets. By this also blood flow which is bleeding out of the blood vessel drastically decreases. Step 3 is blood clotting phenomenon. In blood clotting phenomenon, there are 3 reactions takes place. In the first reaction, intrinsic and extrinsic path results so much amount of the thromboplastin which accumulated in the blood. So by the help of calcium ions, this accumulated thromboplastin converts into thrombokinase enzyme. One should recognize thromboplastin is a protein, thrombokinase is an enzyme. So enzyme is coming from the protein by some factors, so many factors, pro-convertin, pro-accelerating, anti-hemophilic factor, plasma thromboplastin components and all. So once this thrombokinase is formed by the help of elemental calcium and other clotting factors, then this thrombokinase helps to convert the prothrombin into thrombin. This is second reaction. First reaction result thrombokinase. Thrombokinase is the product of first reaction and it acts as a converter or activator from the prothrombin into thrombin. So that result of step 2 or product of step 2 is thrombin. So step 2 product helps to step 3. Step 1 product helps in the step 2. Step 2 product helps in the step 3. Step 3 product helps in the clotting. So this is a very finest way to understand. So one reaction doesn't occur, next reaction cannot be initiated. That is why hemostasis is also called cascade mechanism. Clear? So entire this process should be done within 3 to 8 minutes according to the injury. A small cuts or falls, sometimes some accidents. Bleeding will be stopped within 10 minutes or 8 minutes. Means all these factors are present is the situation. If anyone is absent, the condition leads to hemophilia. Whereas another condition like when in the blood banks, blood will be stored in the bottles and some packets. How it takes place? Because of some sodium citrate, sodium oxalate and EDTA. So all these chemicals helps to store the blood some months or years. So the blood clotting phenomenon is completely different inside the body and outside the body. What you learn is clotting of blood at the bleeding condition or during accidents or traumas, how blood vessel wall which is damaged can be filled completely by some serial of reactions. Practice well this session and attend assignment. Thank you.